matching the following question. So the list 1, they have given the enzymes, and the list 2 will be the functions. So enzyme are histone, acetyltransferase, cyclin-dependent enzymes, alpha antitrypsin, topoisomerase. In the list 2, DNA replication, chromatin remodeling, cell cycle regulation, and MC-RIMA. Okay, so these are the functions and the enzymes. So we need to identify the action of the enzymes. Okay, so the option C will be the correct answer. For histone acetyltransferase, it will be chromatin remodeling. And for cyclin dependent kinase, it will be cell cycle regulation. Okay, CDK enzyme. And for antitrypsin, it is MC-RIMA. And for the topoisomerase, which will be involved in the DNA replication for removing the supercoiling. Okay, and this option C will be the correct answer. And the next match the following question will be, so the list 1, they have given animals like annelids, insects, musculus, and cyclotomes. And the list 2 will be larva stage, veliger, trochopore, ammonocytes, caterpillar. Okay, so we need to match it. So the option C will be the correct answer. So the annelid, it is trochophore. Okay, for the insect, it is caterpillar. And molluscs, it is Velger. And for cyclostomes, it is ammocoids. Okay, and the option C will be the correct answer. The next one is the match the following. The list A has a hormone, and the list 2 will have the functions of the hormone. Okay, so for this question, the option D will be the correct answer. Okay, so the epinephrine hormone in the liver, which will be responsible for conversion of glycogen to glucose. Okay, and the infectious hormone will be increasing the estrogen synthesis, which option it is 3. Okay, and the vasopressin will be reabsorbing the water molecule. Okay, and the epinephrine in the skeletal system will be helping for converting a glycogen into glucose 1 phosphate. Okay, so this option D will be the correct answer. And the next one is the list 1 is this question is also the match the following question. The list 1 will having a nitrogenous basis, and the list 2 is deaminated product. The list 1 is cytosine, methyl cytosine, adenine, and guanine, and the list 2 will having thiamine, xanthine, uracil, and hypoxanthine. Okay, so we need to correctly match it. So the option B will be the correct answer. For cytosine, it is uracil. And for the methyl cytosine, it is thiamine. And the adenine is for hypoxanthine. And the guanine is xanthine. So there we need to match the deaminated product. Okay. So after removal of amine group, what will be the product? We need to match it. Okay, the option B will be the correct answer. And the next one is they have given the compound as well as the function of the compound. We need to match it. The list 1 is diachicol, vitamin K, vitamin A, and warfarin. And the list 2 will have blood clotting factor, antioxidant, anticoagulant, and sugar carrier. Okay. So we need to match it. So the diachicol will have the sugar, it will be acting as a sugar carrier, and the vitamin K is blood clotting factor, and the vitamin E is the antioxidant. Okay, so vitamin E is an antioxidant, and warfarin is a anticoagulant. Okay, so option A will be the correct answer. Okay, and the next one is this one have given the cell type, and the list 2 they have given a function. So the macrophages, what will be it will be going? What action does it go? So it will go for the phagocytosis process, cell engulfing process. Okay, and the B cells, it will go for the antibody production. When it recognizes the antigen, it will convert into plasma cell and memory cell. The plasma will go produces the antibodies. It will circulate throughout the bloodstream. Whenever it finds antibody, whenever it finds an antigen, it will start encountering to it. Okay, so the B cells is responsible for antibody production. And the cytotoxic T cells will be interacting with the infected host. The match will be C. In the list 2, it is 4. C for 4. And the helper T cells will be interacting with macrophages, and it will helpful for the secretion of cytokines. Okay, the option B will be the correct answer. And the next one, There again, it is a match the following question. They have given the scientists and their discoveries. Okay, so Avery and McCall and MC Catty was found what? It will be the chemical basis of heredity. That DNA is acting as a genetic material. They have used the they have done the different kind of experiment to prove the genetic material. Okay, so it is chemical basis of heredity. And the Edward Jenner, it is vaccine. Okay, so they have identified the vaccine and from the cowpox for the smallpox. Okay, and the next one is Erwin Chargaff. It is for love of equivalence. Okay, and that concentration of adenine base is equal to concentration of thymine base. Likewise, concentration of guanine is equal to concentration of cytosine. Okay, so if it is having 50%, that complementary, the pyrimidine base will also in the 50%. Okay, that was proved by the Erwin Chargaff, the law of equivalence of bases. Okay, and Samuel Benzer was the complementation analysis. Okay, so the option B will be the correct answer. Okay, and the next we will move on to the statement related questions. So they have given two statements. We need to identify whether both statements are correct. If there was any incorrect in the statement, we have to select according to the options they have given. Okay, so the statement 1, the application of herbicides to kill unwanted plant is widespread in agriculture. Okay, and the statement 2, DCMU is act by blocking electron flow at the guanine acceptor of the photosystem by competing for the binding site of the plastoguanine. In the light of above statement, choose the correct answer from the option given below. Okay, statement 1 and 2 correct. Both statements are incorrect. Statement 1 is correct. Statement 2 incorrect. Statement 1 is incorrect and statement 2 is correct. So we need to identify with the when statements. Okay, so the herbicides will be help to what? Kill the unwanted insects. Okay, it will also have a side effect of killing the unwanted plants also. Okay, so this is a first statement. It is also a correct one. Okay, and DCMU will be acting as a blocking of the electron flow. Okay, and it at the guanine acceptor by competing for the bind site of plastoguanine. Okay, so this statement is also a correct statement. So the option A, both the statements are correct. Okay, and the next one is in prokaryotes, all type of RNA are transcribed by hollow enzyme that consists of several polypeptides, 2 alpha, 1 beta, 1 beta dash, and 1 sigma. And the statement 2, the start signals on DNA are called promoters and they represent the initial binding site for the RNA polymerase. Okay, so we need to identify whether those statements are correct or not. Okay, so if there were first statement, let us take first statement. So the RNA will have the RNA polymerase is made up of different kinds of subunits. Okay, it will have alpha, beta, and the sigma. Okay, so only with all the subunits it will make as a core enzyme. Okay, so that all type of RNA will be transferred by the hollow enzyme. Once it attached with the sigma factor, it will become a core enzyme. Okay, so the statement 1 is correct. And the statement 2, the promoter will be acting as a signals for the transcription process. Okay, because if the RNA polymerase is going to identify the promoter sequence, once it attached, it will start synthesizing a RNA molecules. Okay, so the statement 2 is also correct. So the option A will be the correct answer.